All right, we're into chapter 17 now. And in this chapter, we have kind of a diverse set of topics. We're first going to talk about vaccines, how they utilize the immune system, particularly the adaptive immune system. This is going to be the main portion that will be included on your paper in chapter 17. The remainder of chapter 17 will talk about diagnostics that use immune molecules and also immune disorders, which includes things like allergies, which we call hypersensitivities, and genetic disorders that lead to problems in the immune system, as well as cancers of the immune system. So there's a lot of what I would say general interest stuff in here that I think will be interesting to you, but not necessarily directly related to the paper. So don't worry if we're going into too much detail. It's more for your own knowledge so that you can understand some of the things that you might be encountering out there at a deeper level. So we're first going to talk about vaccinations, like I said. Then we will get into immunological tests, so using antibodies. We'll talk about those allergies, hypersensitivities, and then we'll get into the issues when we have problems in the immune system. So let's start with a case study that unfortunately is all too common now, I guess, or becoming more common. We have Maureen. She's 56 years old, and she is currently undergoing cancer treatment. She has a sore throat, fever, runny nose. This develops into a very strong cough, and then she has trouble breathing. So her husband takes her to the hospital. We need to know a little bit about her to try and figure out what might be the cause of this. Obviously, she's elderly, so there are more things that could likely be wrong here. Maureen is a kindergarten teacher, and she's been on her cancer treatment, which is, as we'll talk about, a monoclonal antibody for the past 6 months. So she has a B-cell lymphoma. So she has, in her B cells, cancerous cells are growing. This monoclonal antibody binds to a protein on those B cells, and then a group of cells called natural killer cells come in, recognize that antibody, and destroy the cancerous cells, which are more susceptible to this than normal B cells. But that means she is slightly immune compromised because obviously, as we know, the B cells are important in making antibodies. She's been visiting a physician regularly. She has all her childhood vaccinations and adult vaccinations. So hopefully she should be covered. So we're going to do some tests. And first we do a blood count. She is neutropenic, so she has a low white cell count. So something is wrong there. We do a chest x-ray. She has some sort of pneumonia disease. We can see inflammation in the lungs. So the first thing they're going to do is they're going to empirically treat her with an antibiotic. We don't know what is causing this pneumonia yet, but we're going to try something because she's in a very risky place. She's having trouble breathing. So they try a cephalosporin type antibiotic, but as they continue their tests, they find that there are no bacteria in there. So there's no bacterial infection. So obviously we're going to stop the antibiotic at that point. It's not going to do anything. So when we find out that there's no bacteria in there, we have to shift gears and start thinking about other things. One thing is that the next day she develops what we call a maculopapular rash, which is these raised little red kind of bumps here. And this points us towards something that commonly has this called measles. So measles is caused by a virus. So we can start testing specifically for measles using PCR and also checking for antibodies against measles. But surprisingly, even though she has the infection, she's not making any IgG or IgM against the measles virus. So something is wrong here in the production of her antibodies. I think given what her cancer is, you can probably put two and two together. So we have to treat her. We're going to treat her with an antiviral drug called ribavirin and also give her IV immunoglobulins, which help boost the immune system up. We're also going to want to track down where this came from. In this case, it turns out that one of her 4-year-old students was in the incubation phase of measles, and this is before they have any symptoms or anything. So she got the measles from this student. This particular student, along with several others, had not been vaccinated before they attended school, so they were able to carry and transmit this. Now, this is a bit strange because Maureen had her childhood vaccinations and should have been vaccinated against measles. But because of the chemotherapy, this left her immune compromised. She had lower B cell counts, and that left her vulnerable to things that previously she'd been vaccinated against. So that's why we have no IgG or IgM being made, because the B cell count is very, very low. Measles, the virus, is super contagious. We're talking 90% of unvaccinated people who encounter the virus will get it. That's like 9 out of 10 chances that you will get it. So, unfortunately, we are seeing this much more frequently. We'll see some statistics at the end of this chapter, but measles is becoming a problem again. There is a large and growing contingent of people that are choosing not to vaccinate either themselves or, more worryingly, their children. This has been based off a false claim that vaccines are in some way linked to autism. There is copious amounts of data out there that shows that this is not the case. In the early 2000s, there was some poor science done, and some less than reputable people who were pushing this theory, mainly for fame and accolades and things like that. But there is no difference in autism rates between vaccinated and unvaccinated populations. There's so much data out there about this. There is, however, a very significant difference in the rates of disease. If you are not vaccinated, you are far more likely to get a disease than if you are vaccinated. There is no denying that. We have great data from the pandemic of this. At the height of the pandemic, when vaccines were being made available, 98% of COVID-19 cases were in non-vaccinated individuals. Okay, I think that's pretty strong evidence, right, that vaccines were working to shield people from getting a disease. It works just the same with other diseases, and COVID was very serious. It affected the elderly more, so maybe we don't feel that as strongly. I don't know what that says about our society, but when we have childhood diseases, that is something that I think we will start to feel much more as people choose not to vaccinate their children. Their kids are going to get sick. Their kids are going to be crippled and may die. And hopefully we don't need to get to that point to get vaccination back up to where it should be. We'll look at this at the end of this section. First, let's talk about how vaccines and immunizations actually work. We're going to talk about both passive and active immunization. We need to talk about what makes an effective vaccine, and we'll talk about some of the different types of vaccines, and then this key concept called herd immunity. Okay, so if we remember way back to earlier in the semester when we talked about vaccination, you might remember that milkmaids claimed to be immune to smallpox if they had previously got cowpox. Cowpox is a virus that is related to smallpox. It can cause small lesions. Smallpox will cause lesions across the whole body and can be deadly. So, vaccination, our process of inoculating people with cowpox, vacca is cow in Latin, this process led to this theory that if we have inject people with one antigen or one disease-causing microbe, maybe it can protect them against another. This is an example of what we call active immunization. This works to stimulate a person's immune system before they get sick with the microbe. So this could be something like injecting an antigen, which stimulates the immune system, causing antibodies and T cells to be made, but more importantly, memory to be stored. That memory is what protects us when we actually encounter the pathogen. So this is including vaccines. And you will notice in this is injecting antigens. There are various ways we can accomplish this. Some of them work better than others. We'll talk about that in just a second. Passive immunization is something slightly different. This is providing immune molecules to one person that come from another individual. I say from one person to another, but generally that's not the case. We're not sucking immune molecules out of people. A lot of times we use rabbits or sheep or things like this. So it's providing one person immune molecules from some other organism. This could be protective antibodies that are administered by injecting immunoglobulins, like we talked about. Monoclonal antibody therapy would be one example. If we go back to Maureen's case, she was on a monoclonal antibody therapy for her cancer, and she would have also got immunoglobulin molecules to help with the measles. Antitoxins, we can expose organisms to microbial toxins, and they will make antibodies and antitoxins to them. Also, breastfeeding. This transmits IgA antibodies from the mother to the child. So taking these antibodies or immune globulins and moving them from one individual to another. We'll talk about the COVID-19 monoclonal antibody in a later section here. So what makes a vaccine effective? Unfortunately, the, best vaccination long-term is the microbe itself, right? That has all of the antigens, all of the possible things, and your immune system is going to respond to it the best. There are problems with this, and we saw this, debate highlighted during the pandemic. There was a large portion of people that said, I am going to get the disease naturally. That is something people could choose to do. It doesn't sound very fun to me, but it will technically generate the best immune response. The problem is you have to get the disease. Interestingly, we don't have anyone saying that they would like to get something like HIV naturally, right? I bet if there was a vaccine against HIV, people would be very in favor of that, right? So diseases are problematic because they can damage or kill the patient. We saw that in the pandemic, right? Over 1 million people died from it. That's quite significant. So vaccines are building immune response without having to get the disease is the key. So we have a couple key requirements here. Number one is the vaccine should not harm the person being vaccinated. Obviously, that's the whole point right here. You can develop an immune response without being harmed. That's the goal. It needs to stimulate both the B cells, which make antibodies, and the T cells, the cell-mediated responses. If you get both sides of that going on, it's going to provide strong protection. We also want it to result in long-term memory. A lot of the research into vaccines, where they're trying different antigens, is focused on this, right? Both stimulating the immune system and getting that memory, because we want it to last for a long period of time. It shouldn't require too many boosters. This is an interesting thing. Humans are inherently lazy, and the more times they're required to go do something, the less they're going to do it, right? We saw with COVID-19 vaccination, there was like a 10% drop off almost in people who got the first dose and the second dose. That's just generally out of sheer laziness, right? So the fewer boosters you need, the more likely people are to get them. And then obviously it should protect against the pathogen. This is tied into stimulating the B and T cells and the memory part of things. So all of these are things that we look for in an effective vaccine when we're designing them. The key, though, is that people don't get harmed here, right? Yes, you might feel a little crappy after a vaccine, right? Because your immune system has been stimulated, but there is no damage to your body from that. It is your immune system ramping up and getting ready to protect you against that potential pathogen. There are other factors that go into making vaccines. Cost and manufacturing are big problems. That has, in a lot of ways, limited what we're able to make and how much vaccine we're able to make. mRNA vaccines have really changed that. They are much cheaper to manufacture. Storage and shelf life is another one. mRNA vaccines are very hard to store. You have to store them at like -80. They are not super stable, so that is a problem there. We have 4 different basic types of vaccines. 3 of them are much older, I guess, technologies. Actually, they go back to what we talked about with Louis Pasteur. So Louis Pasteur originally was studying chicken cholera bacteria, which can kill chickens. So he was interested in what causes this disease because chicken farmers were losing chickens. He noticed that with his fresh chicken cholera cultures, if he inoculated a chicken, the chicken would die. Okay, so he's doing that, and he's getting ready to go on his summer holiday, right? So he accidentally left out some cultures of bacteria. And then when he came back, he found them out, and he goes, all right, well, I'm just try it anyways, right? Let's see if they're still good. Over time, the culture had lost its potency, its ability to kill the chickens. So he inoculated chickens with this, and they did not become sick. That was interesting. He goes, oh, darn, my chicken cholera has gone bad. I hate it when that happens, right? So he goes and gets a new sample of chicken cholera, and he grows it up, and he injects some chickens, and they die. He then takes the chickens that he had previously injected with his faulty culture and injects them with it. And he notices that they don't become sick. So he hypothesizes that this pre-exposure to the weakened bacteria has actually made them resistant to this sickness. And this is a process that we now call attenuation. Attenuation is one of the ways that we make vaccines. You can start with whole cells, right, and destroy them in various ways. You can heat kill them or inactivate viruses in various ways. That would be dead. Attenuation is different. It's weakening. So let's start with the killed whole cell ones. Okay, so you have bacteria here, and you expose them to heat or radiation, and they die. They're no longer viable. You could still inject those bacteria into an individual. They will not reproduce, which is great because then they can't cause disease, but they still have the antigens on there, those proteins and things that cause an immune response. Our vaccines for cholera and rabies, which aren't super common, but they do exist, they are examples of killed whole cell vaccines. Okay, that's one way. You could also take the live pathogen and attenuate it or weaken it. There are various ways you can do this. You can expose them to heat or other chemicals, phenol, formalin, things like this that weaken them. And they will still grow in the host, but they do not cause disease in the host. So this is actually interesting because they're able to still replicate, but they don't cause disease. So they kind of mimic an infection and actually can generate very strong immunity to this. So we have things like our measles, mumps, rubella vaccine that are live attenuated viruses or bacteria. There's a third kind of vaccine that is a little bit different. Many organisms make toxins, and we can make antibodies against those toxins. So our tetanus vaccine is not actually against the tetanus bacteria, but against the tetanus toxin. That is why you can get a tetanus shot after you believe you've been exposed to tetanus. It's not actually fighting the microbe, it's fighting and destroying the potential toxin that's there. So these tend to be made out of purified antigenic subunits. So little bits of the toxin itself invokes an immune response and bumps up your immunity. Okay, those are the three historical ways that we made vaccines. And these ones take a lot of time, energy, and work to test them, to make sure they're safe, and to grow them and to distribute them. And because of that, they have not changed very much, for good or bad, right? They work, but they're very difficult to make. DNA and RNA vaccines have really completely changed all of this. Working with DNA and RNA in the lab is so simple. It's so easy for us to do. So mRNA vaccines, remember, are little nanoparticles of mRNA that we can inject, and then the RNA will go into your cell, and it contains the information that codes for the antigen, something like the SARS-CoV-2 spike protein. We can adapt these and change these very, very quickly, because all we have to do is change the sequence of the RNA, which we can do in a couple of days, right? And that gives us more time to test them, to make sure they work, to make sure they're safe, but it also makes it much cheaper and easier and more available. So one of our criteria was that it needs to stimulate the immune response. Unfortunately, the best way to do this is using a live attenuated virus. That's because it's live and can replicate and will actually stimulate a bit of the innate immune system. It will stimulate both the cell-mediated and the humoral systems as well in the adaptive system. DNA and mRNA vaccines don't stimulate the innate system because there's no replicating organism in there, but they will stimulate the cell-mediated system and the humoral system, so they are very good. The dead whole cell tend to only stimulate the humoral system, so they are not quite as strong there. This is one of the problems with them, is they just don't work quite as well. By design, the toxin ones, the antitoxin ones, they're only going to work on the humoral system because antibodies are what deal with those toxins. So our best are the live attenuated, but they're very difficult to make. So DNA and mRNA are probably going to become standards. So remember, the mRNA vaccine, let's look at what's going on. We have our virus. It makes various antigens, like, say, the spike protein. We can take the sequence that codes for that protein and make that mRNA. We code it up in this little lipid nanoparticle that will fuse with our cell membranes. The mRNA goes into the cell, it gets translated by our ribosomes, and we make these little spike proteins. These proteins go on our cell surface and they stimulate an immune response. They don't hurt us in any way. These spike proteins are not the part of the virus that causes damage to us, but they train our immune system to recognize them. So we build up antibodies. We also get cytotoxic T cells that get made against these things. These antibodies levels go up. So when you actually encounter the virus, you're pre-primed and ready to destroy it quite quickly. So there's some great advantages here. They're super quick and easy to make. You don't have to grow any organisms in here. You can change them quite rapidly. They will present to the CD8 cells, and that gets the cytotoxic side working, as well as the humoral side with the antibodies. And antigen presenting cells can pick up these bits and display them to the immune system. The problems, though, are mainly kind of technical issues. RNA itself is not very stable, so the shelf life isn't super long. Once you thaw it, you have to use it. Getting the mRNA into the cell was actually the tricky part. We knew that this was possible for a long time. It was the manufacturing of these lipid nanoparticles and packaging up basically the mRNA that was the real challenge in this. They are technically potentially weaker than a very well-designed attenuated strain. I would say that is completely overridden by the time that it takes to make them. Attenuated strains take a long time and a lot of testing. There is the potential for things to go wrong there, right? Because if you don't attenuate it fully, it could cause disease. The mRNA vaccines, so quick to make. We can do more testing because of that. And I think the biggest thing was that they're new, right? People are scared of new things. People are scared of things they don't understand. There was a lot of pushback to it because people were like, wait, you're going to inject me with like virus DNA or something? Is that going to make me a virus or get me sick or, things like that? If you don't understand it, it sounds kind of scary. So that was one big thing. I think most people are very comfortable with this now. So if you're interested, there are lists here of several of our approved vaccines. There are things like cholera and plague that do have vaccines, but they're not widely distributed. If you're going to like an endemic region, you might get them. Some of them, like the cholera one, it's like short-term protection. Typhoid fever, the oral one is very low effectiveness. So we have some that are only given in certain circumstances. Then we have like the common childhood vaccines. So let's jump over to look at that. So here's a common childhood vaccination scheme. And you'll notice that we don't actually really start to vaccinate until after 2 months of age, because there can actually still be maternal antibodies in a newborn. And those maternal antibodies actually can dampen the immune response of these vaccines. So we got to wait until those are gone before we start vaccinating here. Unfortunately, some of our vaccines we can't give until much further down the road. Something like MMR, measles, mumps, rubella. We don't do that until 12 months of age, or chickenpox. That leaves a whole year where a child is vulnerable. We will talk about how we try to protect those individuals in the next slide. So you'll see here there are some that are becoming more common for older individuals, ages 11 to 12. We have meningococcal meningitis vaccine and things like we talked about Gardasil for HPV. That's one that we don't do until a little bit later, until there are risks of getting these things. Okay, so how do we protect those newborns? Well, hopefully we can do it through a process that we call herd immunity. And this can protect not just newborns, but also people who are unable to get vaccinations or immunizations due to various reasons, or people who are immune compromised. Okay, so herd immunity is when you have a large portion of the population that's immunized, it will actually protect unimmunized people because it cuts routes of transmission. Most estimates put this at about 75% of the population needing to be immunized for this to work. It's going to vary by pathogen and transmission route. Let's take a look at what's happening. Okay, in this example, blue individuals are healthy, unimmunized individuals. Yellow are healthy, immunized individuals. And red is non-immunized, sick, and contagious individuals. So you can see in this population, I don't know, there's about 30 people, maybe let's say, and 2 of them are sick and contagious. The rest are not immunized. We see quite quickly that a large number of people get infected, right? That makes sense. It spreads throughout the population. Now let's jump up to, I don't know, this is maybe a little over 15% of the population is immunized, right? So we have 6 individuals in our hypothetical 30. You can see that we still have those 2 sick people. There are still plenty of routes of transmission for it to occur. So the immunized people are protected themselves, but there is no cutting down of transmission, right? Because this person can't give it to that person, but they can still give it to that person, who can give it to that person, who can give it to that person, who can give it to that person, right? There's still plenty of routes of transmission. When we get to high levels, and this is probably somewhere around 75%, 80% of individuals that are immunized or vaccinated, they shield unimmunized people. So here, right, this person can't transmit to them or them or them or them. Maybe they transmit to that person, right? But we don't have this spreading of the disease through the population. And we still have people who are unimmunized who are protected or shielded by the herd immunity. Okay, you might say, well, whatever, screw them, right? Like, this is part of our society, right? We have to make choices to help people. And we have plenty of examples of choices that we all have collectively made that, I guess, limit our freedoms or enforce rules that limit our freedoms for public health. Let's take drunk driving laws. We have collectively decided that we don't find it acceptable for people to drink and drive. We think that that is potentially harmful, right? Because alcohol dampens the senses and potentially leads to higher crash rates, which could kill someone else. It's kind of the same thing here. This broad vaccination and asking people to make the choice to be vaccinated is kind of like that, because we can protect other people, either who don't want to get vaccinated for whatever reason, or who can't get vaccinated. That's the bigger thing. There are people who have compromised immune systems. Vaccines will not help them. Think about back to the beginning of this chapter when we talked about Maureen. She'd been vaccinated, but she has a cancer that's affecting her immune system. She got measles because someone chose not to vaccinate their child against the measles, and it transmitted to her and could have potentially killed her. So this herd immunity is something that goes beyond just protecting yourself to also helping the rest of society. Unfortunately, COVID vaccine numbers in Idaho quite low. It's about 52% have both doses. In Bonneville County, it's a little bit higher, 58%. That's kind of balanced out because now most people have been exposed to it. So those who didn't get vaccinated probably got COVID. So they're immunized against it at this point. So we're starting to, get to this point, not through vaccination necessarily, but through people having been exposed to it. But it's not perfect, right? We're still seeing cases out there. Let's look at the best case study of herd immunity, though. In 1954, there were 4,000 cases of measles for every 1 million unprotected individuals. So every 1 million unimmunized individuals, we saw 4,000 cases of measles. Fast forward to around now, we have a 90% vaccination rate. So you would say that should cut the number of cases down by 90%. But actually what it's done is it's cut it to well over 99.99%, right? We see less than 1 case of measles per million unprotected. There are still 31 million unvaccinated individuals in the United States. We should see larger numbers than we do. That's because of herd immunity. Because we have such high vaccination rates, even those unprotected, unvaccinated people are protected through herd immunity, right? So whatever 90% of this is much smaller than that. So we're getting overprotection through herd immunity. That's awesome. Also, let's look at data about disease and vaccination. Here is time, so that was 1954. Our vaccination program started in the early 60s here. So we're seeing thousands of cases here, 700,000 cases in 1954. Right, that's a lot. There were almost 800,000 cases of measles in 1958. When we started vaccinating, in a matter of 2, 4, 6 years, we went from 400,000 cases to less than 50,000 cases. And that number has dropped to probably less than 1,000. Here we see in 2010, there were less than 400 cases indeed. But for whatever reason, people have stopped vaccinating. I don't want to get into why they choose to do this, right? There are various reasons, but we are seeing a comeback in measles. In 2019, it was estimated there were about 1,600 cases, which that compares to whatever. That's probably like 10 cases right there, right? That's a huge increase. And if people continue to not vaccinate their children, we're going to see that go up, and that can transfer to people who are unable to get vaccinated. There are occasionally issues with vaccines and allergic reactions, but it's extremely rare, right? There are no links to autism, diabetes, any other disease through vaccines. There is a link to measles if you're not vaccinated against measles, though. So, unfortunately, if we stop vaccinating, the long and short of it is these diseases will come back, and we're seeing that right now. Now, I want to leave you with a very scary example. In the early 1900s, there's a disease called polio, caused by poliomyelitis virus. This leaves children, primarily affects children, it leaves them paralyzed. It can paralyze different parts of the body, so these children all have paralyzed legs, so that's why they all have to walk with crutches. If it affects higher up the spinal cord, it can leave children unable to breathe. They are paralyzed, and they have to be put in what's called an iron lung, which pulls a vacuum, which basically inflates their lungs and deflates their lungs. So we had to stick them in a giant metal tube to keep them alive. Polio has been almost entirely eradicated in the world. We don't see this anymore. I think you can all agree that that is a good thing, right? Children that are not paralyzed, in my book, that's a good thing. We don't want to see iron lung wards where there's just row upon row of iron lungs with polio children in there. We don't want to see that come back, okay? So this is my plea to you. Please encourage everyone you know to make sure that these things don't come back, to make sure that children don't have to suffer for whatever reason. So yeah, I'll leave you with that. That is immunization. We talked about some different types of immunization, vaccination in there, the different types of vaccines, what we're looking for them to be effective. And then hopefully I've convinced you that this idea of herd immunity is something that we should shoot for because it can protect not only ourselves, but our population as a whole. All right. In the next sections, we'll talk about some diagnostics and some diseases that affect the immune system.